students and learners of the Postgraduate Diploma in Environmental and Occupational Health. Today we will discuss in this session for the course code MEV 003, that is Environmental Law and Management. We will discuss on biosafety part 1. Now we have seen that people who work with biological agents, for example bacteria, viruses, or they can even include certain protozoal agents. So some of them may be pathogenic in nature and some may be non-infectious, some may be infectious in nature. So depending on this, we can categorize the biosafety levels and how these organisms, these microbes especially, how they need to be contained properly and how they can be even safely managed in the laboratories. So this session will deal on that aspect, that is biosafety part 1. So after listening to this module, you will be able to define biosafety, you will be able to understand the various concepts and principles related to biosafety, and we will see what are the guidelines that are issued by the World Health Organization and also the various biosafety levels which are involved. Microorganisms can be harmless, they can be beneficial, and they can also be harmful pathogens. So now you would have seen that the microorganisms, they are used even in your own homes for domestic purposes, for example for making curd or for making your dosas and your idli batter. So these are all the useful microorganisms because they are being usefully employed even in the same way in the fermentation of certain food products and also certain alcoholic beverages. So here again the microbes can play a very important role in their advantages for their usefulness. But sometimes the microorganisms, they can be harmful and they can cause several kinds of infection, not only to the human beings but also to the plant flora and to also the animal kingdom. So some non-pathogenic microorganisms, I gave you an example in the processing of the curd, that is milk to curd, but they can also be used in biotechnology to produce enzymes. They can be used as biofertilizers in the agriculture, and let me tell you, these are the safe and the green ways and eco-friendly, environment friendly, pro-nature kind of techniques which are used in the agricultural sector. Same way probiotics, they are used also in the bioremediation of certain places and also for the fermentation in food production and technology. So therefore microorganisms, we can categorize them as harmless, beneficial and harmful pathogens. Infectious or pathogenic microbes. These are a cause of concern both for the animal health, for the human health and also for the plant kingdom. So they have the potential to cause diseases with tremendous economic and environmental consequences. Viruses also are a concern for environmental as well as human health. Pathogenicity. Now when we define pathogenicity, that means it is a capacity of certain microbial strains that has the ability to cause disease. It represents a form of versatility that enables certain microbes to replicate within a specific host, and it can also damage the host cells. The outcome of the infection. Now this is dependent on several factors, that is, it depends on the properties of the pathogen itself, for example the virulence, the invasiveness, toxic or allergenic effects. It also can depend on the host's immunity, like how our immunity is working or functioning. Accordingly, the pathogens can be classified into two basic types, that is the primary pathogens and the opportunistic pathogens. Now primary pathogens, these are those that cause disease among the healthy individuals. Opportunistic pathogens, these are those pathogens which can cause disease only in those people or individuals who are compromised in their immunity. Some etiological agents, they have been extensively cultured and studied. You would have seen there are special laboratories and special working areas where they deal with the infectious microorganisms, and you can even if you have isolated certain strains, you can store them in such places. So the consequences of working with such agents, those people who are working with such agents, they will be getting at some point of time with some lab associated infections if they are not taking care of their health properly. So these infections or such kind of infections, they have been documented as early as the 1890s. Now the people who are working in such the culture sectors, they are at a higher risk for infection with certain very infectious microbial agents. For example, those that are causing tuberculosis, that is Mycobacterium tuberculosis. The same way for the Brucella and also hepatitis B viruses. So these are all very infectious, and those people who are working in such areas, they have the probability of getting infected with the same if they do not protect themselves with the proper biosafety cabinets and also the personal protective equipments. Biological safety. So now we come on to understand what is the meaning of biosafety. So this is new to the field of occupational safety. Developments in genetic engineering, they have influenced the people involved in biomedical research by increasing their awareness of the safety aspects related to their work. Now the most important development with an international impact, this was initiated in the year 1979 by the World Health Organization, and they set up a working group within a special program, and that actually basically dealt on the safety measures in microbiology. Now this working group, they formulated a set of minimum standards for the laboratory, and that was compiled in a laboratory biosafety manual. This manual is very important, and it describes the definition of the risk groups. Mainly we have the 4 risk groups based on the relative hazard of the infective microbes to the laboratory workers, the community, the livestock and the environment as a whole. Now the World Health Organization recommends each country to outline their own classification by risk group of the agents encountered in that country based on some factors. So what are these factors? They are the pathogenicity of the agent. Then they involve the mode of transmission and the host range of the agent. Now this will include the immunity of the people, the host population, the nature of the host population, and also the environmental health and sanitation of a specific area or a geographical location. Availability of the effective preventive measures. So here this deals with the prophylaxis, either it could be using the anti-sera or by vaccination. Then the sanitary measures like the food and the water hygiene, the control of the animal reservoirs or the arthropod vectors, the movement of people or animals, and the importation of the infected animals or the animal products. So these are all taken into account. The final point, there is a 4th one, is the availability of effective treatment. This includes passive immunization and post-exposure vaccination, antibiotics and the chemotherapeutic agents for the possibility of emergence of the resistant strains. The WHO also underlines that the conditions prevailing in that specific geographical area in which the microbes are handled, that should also be addressed while assessing the various criteria for the classification. And in the same way, the individual governments, they should also decide to stop or to prohibit certain pathogens from being imported, from being handled other than for the urgent diagnostic purposes. So they are also referred to as the quarantine organisms. Now let us define the term biosafety. So biosafety refers to reduce and eliminate the potential risk arising from biotechnology and its products. It has also been defined as the avoidance of risk to human health and safety and to the conservation of the environment as a result of the use for research and commerce of infectious or genetically modified organisms. Containment. Let us now define the meaning of containment. This refers to a series of safety measures and methods for managing the infectious agents in the laboratory. Now basically the purpose of containment is to help reduce or eliminate the human and environmental exposure to the potentially harmful infectious agents. Biohazard control. This is another definition which is of importance, and that includes the management of biological hazards through the proper application of engineered containment and administrative controls. Now when we talk about containment, this word appears to be a little bit general for us now, because we have all seen in several epidemics and several pandemics, the health officials use the word containment areas. So from this itself we should understand that what are the safety measures that are used in order to manage such infectious agents, because it could be the infectious agent could be transmitted from human to human transmissions and also from the animal to the human transmissions. So therefore, containment is a very important terminology in biological safety. The types of containment. So there are two levels of the biological containment, and they are the primary and the secondary containment. Primary containment. This protects the people and the immediate lab environment where scientific experiments are carried out from exposure to highly infectious agents. Safe and good microbiological techniques coupled with good safety equipments, they can provide sufficient primary containment. Some examples of the primary containments, they include the safety equipment such as biological safety cabinets where the people work, and also we have the safety centrifuge cups. Now in certain cases when it is difficult to work in the biological safety cabinets, then we have the PPE such as there is a personal protective equipments such as laboratory coats, mask and glove. They can all act as a primary barrier between the personnel, that is the person who is handling these infectious agents, and the infection material. Secondary containment. This is concerned with the protection of the environment, and that is external to the laboratory from exposure to the infectious materials. Some examples of the secondary containment or the barriers, they include the work areas that are separate from the public areas, decontamination facilities, hand washing facilities, special ventilation systems and also the airlocks. Elements of biological containment. Now this includes the safe laboratory practices, good safety equipments and the facility design. These are very important elements. To ensure the minimal exposure to the infectious agents, the workers and the employees, they need to assess the hazards and their occupation also, the work that they are working for. All this they should analyze this well before, and then they should apply the biosafety principles accordingly. Now let us come on to the biosafety levels. Now in this triad or the triangle, you can see the low risk microbes and the high risk microbes. So from the biosafety, we have the biosafety levels 1, 2, 3 and 4. This was established by the Center for Disease Control and the National Institute of Health, United States of America. They are the combinations of safe laboratory practices and techniques, safety equipment and also the facilities. All the biosafety levels, they are most appropriate for the biohazards and for the laboratory activities. So in this, if you see the lowest one, that is the low risk microbes, they come under the biosafety level 1. Then when you go on to biosafety level 2 and 3, we have a little more microbes which are of a certain amount of risk to the human beings. The bio level 4 or the biosafety level 4, they come under the high risk microbes. Now let's come on to the biosafety level 1. This refers to the practices, the safety equipment and the facilities which are appropriate for work with the defined and characterized strains of viable microorganisms which are not known to cause any disease in the healthy adult humans. So for example, these organisms, you can work with them even on an open bench laboratory where you don't have special cabinets or you don't need to have special safety procedures. Just a simple bench which you have in all the laboratories, they include certain types of the normal strains, like it could be the bacillus strains and so on. So here in this figure you can see that this laboratory is separate and scientific work can be conducted here. They can be conducted on the open bench tops using standard microbiological practices. So in this level, as I said, no special containment equipment or any facility design is required. Normally in any bacteriology or the microbiology laboratory, you will have 2 or 3 of the working spaces. So you always have an outer laboratory where there are open benches where the normal procedures are carried out. Then you have an inner region where you have the certain other special facility and design equipments where certain infectious bacteria or the other kind of the organisms are being used for study. And then you have special other certain equipments also like anaerobic chambers and so on, and also certain other protective facilities where only the well trained staff can actually enter those sections and then they can carry out the desired experiments. Therefore, the laboratory personnel should have a specific training in the procedures at every level also that are conducted in the lab, and they need to be supervised by a scientist with a general training in microbiology or in a related science or with even a specialized training in that specific equipment that they are dealing. Further, a biological safety cabinet will not be required for working with these microbial agents belonging to the biosafety level 1. Biosafety level 2. So this means a little higher, you are going above the first level. This refers to the practices, the safety equipment and the facilities that are used for working with the indigenous moderate risk microbial agents, and they are associated with a certain amount of human diseases. Here the lab staff should have a specific training in handling such pathogenic agents. They are instructed by the proper scientist, and the access to the lab is limited when the work is being conducted. So here again you will have certain codes, and only the scientists will be allowed to enter there who are properly trained and who are able to assess the situation and work with these organisms. So extreme precautions are taken with the infectious agents, and certain procedures are conducted in the biological safety cabinets. Biosafety level 3. This refers to the practices, the safety equipment and the facilities that are used for working with indigenous agents with a potential for respiratory transmission that can cause serious and potentially lethal infections. The primary and the secondary barriers are highly necessary over here in this level. Only then the personnel or the person who is working with these infectious agents, they will be protected, especially in the contagious area, the community and the health from exposure to these potentially infectious agents. So the barriers are highly important. And here again a class 2 biological safety cabinet is required for working with these agents. We will discuss about the biological safety cabinets in the next session. The biosafety level 4. This refers to the practices, the safety equipment and the facilities used for working with the dangerous infectious agents, and they pose a risk of life-threatening disease. So this is a 4th level, and it is having a risk of life-threatening and infectious diseases. The lab staff here, they need to have specialized skills and training in handling these extremely hazardous infectious agents, and the use of primary and secondary containments and also special laboratory design characteristics. The lab staff are also supervised by the competent scientists who are trained and experienced in working with these dangerous agents. Access to the laboratory is strictly controlled by the laboratory director. Now this facility is either in a separate building or it could be controlled area. It could be situated in a controlled area within a building which is completely isolated from the other areas of the building. And in this case, a class 3 biological safety cabinet is required for work involving such infectious agents. So you have seen in the special pandemic which occurred now, that is in the recent pandemic, that is the coronavirus. Here again how the people were contained, the containment facilities and all these biological safety cabinets, the personal protective equipments that were used, and where even the buildings where the infectious agents were getting spread, they were all completely sealed, and even there were special scientists who had to carry out the special experiments to understand the virulence, the pathogenicity of these viruses that were replicating, and it was spreading just seconds to seconds. People were actually getting infected, and this was a really highly infectious state and pandemic which occurred and infected a lot of people with multi-organ failures. Now this is a table which is giving you the summary of the biosafety levels and the infectious agents. Now we have categorized this according to the biosafety levels 1, 2, 3 and 4. The first one, if you see, the level of the potential of the infectious agent. So this is not reported to cause any disease in the humans. Some examples of these are the Bacillus subtilis, E. coli and also certain other less infectious microorganisms. Then the techniques that are used, you need just standard microbiological practices, and the safety equipment, we do not require any special safety cabinet required over here, and you can even conduct the experiments on the open benchtop, and a sink is required in order for the hand washing and for cleaning of the normal petri plates and whatever is being used. The biosafety level 2, which is reported to cause a moderate amount of infection. This is known to cause human disease, percutaneous injury, mucous membrane exposure, etc. Some examples are the measles, hepatitis B virus, Toxoplasma species and also Salmonella species, which causes typhoid. Here the techniques used, which include certain sharp precautions are required. The sharps that are used, for example the needles or the type of the medical sharps that are used. Then the biosafety manual is very much required, so that we can understand that, and we need to see how these biomedical waste also, whether they can be decontaminated, how that has to be properly disposed, and decontamination of certain decontamination procedures, the medical surveillance policies, and also respiratory protection, how much is required. So all these protocols need to be taken into account. The safety equipment, here we will require a class 1 or a class 2 biological safety cabinet. Then the physical containment devices, that is used for agents that cause the splashes, the aerosols, infectious materials. Then you need the lab coats and the gloves, the face mask, and also a complete apron or a gown, which is personally protecting the person. Now the facilities required are the level of 1 plus, that is an autoclave is definitely required over here. The third one, that is a biosafety level 3, these are the indigenous agents with the potential for the aerosol transmission. So they are transmitted easily in the air from one person to another. The disease may have serious or lethal consequences, and it also depends to a certain extent on the host immunity. Then the examples are the Mycobacterium tuberculi, you have the Coxella, then you have the Bacillus anthracis that causes anthrax, and certain other viruses that causes encephalitis. Level 2 plus, this is referred to as, and here again decontamination of the waste is important, decontamination of the lab clothing, then the baseline serum samples of the lab personnel, all this has to be procured and taken out properly. Now primary barriers are required in this case for the level 3, the class 1 or the class 2 biological safety cabinets and other physical containment devices that are used for the other open manipulations of the agents. Here again all the personal protective equipments are required, like the lab coats, the lab clothing, the gloves, the respiratory protection, goggles and everything. Level 2 plus. So here again we have the physical separation from the access to the corridors. Then you need the self-closing doors and the double doors, so that again more barriers are required for less transmission of that agent from one room to even another. Then the exhaust air, that also needs to be taken into account, that the same air is not getting recirculated within the area or the room. Then there should be a negative air flow into the laboratory. The biosafety level 4. So dangerous and exotic agents that pose a high risk of life-threatening disease. For example, the aerosols that are transmitted from the lab, or infectious or related agents with unknown risk of transmission. Some examples include the Ebola Zaire. Then you have the Rift Valley fever, those infectious agents, and also the Nombre virus. Level 3 plus. Here again the clothing has to be changed before entry into the lab. Shower when you exit from that place or where you are dealing with these infectious agents, a shower is definitely required. All the material has to be decontaminated on exit from the lab. All the procedures that are conducted in the class 1 or the class 2 biological safety cabinets in combination with the full body, which is air supplied, positive pressure personnel suit, these all need to be taken into account. Level 3 plus. Here again a separate building or isolated zone is required. The exhaust vacuum and decontamination systems are required. Additional requirements that are outlined in the biosafety and the microbiological and biomedical laboratories manuals. These definitely have to be taken into account. They need to be read, well read properly, and a scientist who is supervising all this needs to be well trained regarding these aspects. So my dear students, in this session we have understood the terminologies, the definitions related to biosafety, biological hazards, infectious agents, containment, which includes the primary and the secondary containment, and the different biosafety levels, which included from 1 to 4, and how infectious and how less infectious an agent can be, and how the personnel or the people who are working in such laboratories need to be well protected, both in terms of the laboratory equipments, the laboratory devices, and also the personal protection equipment, and how well trained they need to be in order to handle such infectious agents. Thank you for your patient listening.